Bismillah ar-Rahman ar-Rahim. By the name of the God, with you, Professor Dr. Khaled al-Musallami. Khaled al-Musallami. Zagazig University, present in Egypt. Medical School. My watts is 0122741935. I will start with you the study of the histology. What means by histology? Histology, it's two words. The first word is histo, tissues. The second name is, or our second part of the word is ology. It means the science. We can mix them in one word, in the one science, which are called histology. Is the science which study the microscopical structures of the normal tissues, both lying under normal, both lying under microscope. But this is the science which study the normal, not diseased. Why? Second branch called pathology, which study the disease. To know its tissue is diseased, must know the normal structure. Normal structure I want to know. I want to study. To told you that is the other tissue is diseased. So the histology, it's very important for medical student, for pharmacy, for dental, and for veterinary, and for anyone study in medical department or any medical part. To make this, I must told you that is the tissues. Our tissues, when I will take piece from your tissue, from any part of the tissue, at the first, before start, that is what I will told you, that is our body formed from many organs, many systems, respiratory system, digestive system, urogenital system. Each one of them formed from organs. Each organ formed from tissue. And each tissue formed from cell. So our study around the cell, around the tissue, under microscope. From the basic knowledge for you, I will told you that is the tissues, or when I take any piece from our bodies, it's very colorless, no color. I take from the hand, put under microscope, no color. I take from the ovary or liver, I told you it is no color. It is colorless, no color in our tissues. So when I put them under microscope, I cannot see. I cannot see anything. Why? It's colorless. So I must stain the tissues with basic stains. I will stain the tissues with basic stain. The cell formed from two parts, cytoplasm and nucleus. Cytoplasm and nucleus. Cytoplasm. I will normal, no color. I put here stain to stain the cytoplasm with red color. Which is the stain? The cytoplasm. I give two stains. One of them called eosin, the second called hematoxylin. Eosin have, that is E, and hematoxylin H plus X. The eosin stain is red in color. Hematoxylin eosin blue in color. that is blue, that red. Chemical reaction for eosine, it is acidic. Chemical reaction for hematoxylin, basic. When put them on the cell, the cytoplasm takes the stain which is called eosine, which is acidic. So it is alkaline. The cytoplasm is alkaline. And he like the acid stain. So I will give name acidophilic. What does mean philic? It is like what? Acid, which are eosine. That is the normal basic structure of the cytoplasm when stained. But the nucleus containing which is called DNA. DNA. What is means by DNA? Deoxyribonucleic acid. So the nucleus containing acid. Why containing acids? It contains DNA. When put the stain here, it will react hematoxylin with the nucleus, stains the nucleus. So the nucleus is stained by blue color. Though the nucleus like the basic structure, so I give the name for the nucleus basophilic. Basophilic. What means by basophilic? Baso, basic dye, which is hematoxylin. Philic, like dye, which is hematoxylin. So I will, in the light microscope, I take the two colors. Red color for the cytoplasm, blue color for the nucleus. Why is the cytoplasm takes the red color? Because it can react with a acid stain which is called eosine. Eosine red in color. So the cytoplasm appears red in color and gives the name acidophilic. The nucleus containing DNA which is acid, and this is will react with hematoxylin which is basic dye blue in color. So the nucleus take the color blue in color, so I give name basophilic. That is the common stain, hematoxylin and eosine. So I will here in the structures told you why the cell is acidophilic, why the cell is basophilic. Acidophilic like acid, basophilic like base. So the normal structure or normal reaction in the cell, the nucleus is containing acid, it is acidic, and the cytoplasm alkaline. That is the light microscope. Such stain. The second microscope will use its electron microscope. In electron microscope, I will use the electrons, but no color. No color. Why? That is present in EM red? No. That is present in the nucleus blue? No. Which color will appear in the electron microscope? It's white and black. White and black. Because in simple words, the electron microscope one minute. The electron microscope will give the light similar to give electrons similar to light. That is the way of electrons. That is the tissue. I told you it's colorless. When put in the way of light, I saw the black image. That is the black image, no color. That is the structure of the organelles, structures of the cell appears black. When the electrons descend, it's fixed in the tissues, and the image of the tissue will appear here on the screen. So it's black in color, no colors in electron microscope. When colored the tissues appears in color as here. I want to black. Yes, that is black. That is, it is appears dark line. Dark line, why? Take electrons. Dark line, why? Takes electrons. That is called electron dense. Take many electrons. And that is the line appears not black, it's appear white. That is called electron lucent. Takes few electrons. So it is appears here white, but here take many electrons, it appears dark. That is the introduction for histology. I told you the name of histology comes to study the normal tissues under microscopes and any types of the tissue, normal tissues. And I can use the two types of microscope, light microscope, electron microscope. In light microscope, the tissues appears colored red and blue, but in electron microscope, dark and lucent, dark cold electron dense, and the light cold or gray in color called electron lucent. We can study the tissues, the cells in different lecture, and the next lecture will tell you the structure of the histology for all our tissues, our body. Thank you very much, ladies and gentlemen, with Professor Dr. Khaled Hamid El Misallami. Thank you very much.